Hello and welcome to another lecture of the course Aquaculture. Today we are going to discuss about culture method of giant freshwater prawn Macrobrachium rosenbergii, and in today's lecture we shall be discussing about biology of Macrobrachium rosenbergii. The giant freshwater prawn Macrobrachium rosenbergii is widely distributed in tropical and subtropical countries, in almost all the major countries which are doing aquaculture, um, including our country, India, Bangladesh, Burma, China, Philippines, Thailand. All these regions, the giant freshwater prawn Macrobrachium rosenbergii is distributed. Now, morphology of giant freshwater prawn Macrobrachium rosenbergii, a little bit. Um, I could have given you a reference in my earlier lectures, but here you can have um, relook at that. Body covered with hard shell. We all know about these prawns. The post larvae and adult prawns have the head and tail as cephalothorax and the abdomen that we all know. Body divided into 20 somites or segments. A head consists of 14 segments which fuse together, and they are in fact not visible directly, but they are under a large dorsal and lateral shield that we call as carapace. This is uh, the structure that is visible and that provides shelter to or protection to the animal. Rostrum is slender and curved upwards. That is one of the important points of these prawns. The tail is very clearly divided into 6 segments, each bearing a pair of appendages known as piliopods or swimmerets. So, these are the main morphological features of the Macrobrachium rosenbergii, the giant freshwater prawn. Here you can see that in the diagram or picture. The rostrum part, cephalothorax, then the swimming legs that have been shown, telson, uropods. Then here or in this one also you can see that they have uh, you know marked labeled as uropods, telson, antenna, then swimmerets, pleura, and carapace, of course, of the different body parts of morphological features of giant freshwater prawn Macrobrachium rosenbergii. The color of the body is tends to be brighter in younger animals and generally darker and bluish or brownish in case of older prawns. Post larval prawns are usually having a greenish brownish gray coloration, whereas that sometimes you can find as bluish with an irregular brown or gray and whitish longitudinal streaks on the body. Orange spots may be visible where the tail segments bend. You can find the orange spots. Mature and male prawns are considerably larger than the females. Here, in the case of giant freshwater prawn Macrobrachium rosenbergii, males, when the full grown males, adult males, mature males, you will find them larger in size compared to females. Identifying characteristics: very long rostrum with 11 to 14 dorsal teeth and 8 to 10 ventral teeth. Tip of its telson reaches distinctly beyond the posterior spine of the telson. So these are some of the identifying features, characteristics. Again, a photograph of Macrobrachium rosenbergii that is shown. Now, habitat-wise, the giant freshwater prawn or Macrobrachium rosenbergii is showing nocturnal behavior, bottom dwelling, and sluggish in nature, and it shows territoriality. During the day, they remain half buried in sediment and prefer shallow, detritus rich, and vegetated areas. They are found in most inland freshwater areas, including lakes, rivers, swamps, irrigation ditches, whatever kind of aquatic habitat, freshwater habitat they can approach to, they are distributed, as well as in estuarine areas. The depth usually at which they are found, it may be 0 to 2 meter, because they are bottom dwelling, so even they can be found even at a very low depth. Gravid females, egg and larvae, they inhabit in estuaries or brackish water. Post larvae and adult, they are bottom dwellers and found in fresh waters. Feeding habit, larvae mostly consume zooplankton, mainly minute crustaceans, very small worms and larval stages of other crustaceans they can also consume. Post larvae and adults are omnivorous, eating algae, aquatic plants, mollusks, aquatic insects, worms, and other crustaceans, and that is the probably the reason of their diversified distribution in the PL and adult stages, PL and beyond. Here it is shown the life cycle of rosenbergii. Here, if you start from here, eggs carried by varied females. These varied females are found in freshwater, and number two, these eggs are carried by varied females. Number two, eggs hatch into larvae in brackish water. For their breeding, for larval development, they need brackish water. So that is why you have seen that these stages are distributed in brackish water. And third point is larvae metamorphose to post larvae. Then larvae are metamorphosed into post larvae, and these post larvae now develop into juveniles and are distributed in fresh waters, and they will be there until adults. And when again they have to breed, then they will move to brackish water area, because larval development will occur in brackish waters. Spawning and fertilization. Sexually mature female prawns undergo a pre-mating molt. That is one of the characteristics of these prawns, that molting, pre-mating molting or pre-spawning molting is noted usually. Molting is shedding of the exoskeleton or outer shell of the growth that we all know. The male protects the female undergoing pre-mating molt from the other females, as they are prone to attacks. Courtship involves cleaning stage, where male clean the ventral region of the female for laying the eggs. During copulation, what happens? The males release its spermatophore, a gelatinous mass containing sperm, through openings, or what we call as gonophores, at the base of the fifth working leg. After copulation, the female will release the eggs through openings at the base of the third working legs. The fertilized eggs are deposited in the brood chamber, which is the expanded abdominal area of the female, right? So the males release gelatinous mass containing sperms at the base of fifth working legs. After copulation, the release of eggs, female will release the eggs through openings at the base of third working legs, and the fertilized eggs are deposited in the brood chamber, which is expanded abdominal area of the female. So these three major steps during spawning and fertilization. And here you can see one photograph has been shown where courtship is going on between this blue claw male and the female giant freshwater prawn. Eggs laid by females resemble like berries, as they held together by a sticky substance. And this is important, eggs laid during first year of maturity is 5,000 to 20,000 in number, whereas when they remature at later stages of their life, then the eggs may increase in number significantly, like 80,000 to 1 lakh eggs can be produced by a female when it rematures. The different life stages of Macrobrachium rosenbergii. The embryo eggs of the prawn are slightly elliptical, the longer axis being 0.6 to 0.7 millimeter in length. Embryonic development starts as soon as the eggs are extruded by the female. Average time for hatching is 20 days. They are In fact, this one is interesting. You will get to see this if you visit any of the Macrobrachium rosenbergii hatchery. They are bright orange in color until 2 or 3 days before hatching. As soon as they start changing the color to slate gray, that means the hatching point, hatching time is approaching. Some stages of egg development of the giant freshwater prawn. A is showing completion of segmentation, 24th post-fertilization, 24 hours post-fertilization. B is showing eye pigment developed by day 9 and larva fully formed at day 14 in case of C. The first nuclear division may be observed about 4 hours after fertilization, and all these timelines, they are dependent on many other factors like temperature can have a strong influence on these timings, then developmental timings, and water quality may also have influence over these timings. So there may be variation that you can observe when you are running Macrobrachium rosenbergii hatchery, but you will have to standardize all these parameters. The time taken timeline you can rewrite or standardize and rewrite for your own region, for your own area, for your own hatchery. The cleavage is completed in about 24 hours. Temperature optimal range is 26 to 28 degrees centigrade. Optimal salinity is 6 ppt. High temperature should be avoided. So if it goes more than 30 degree, it becomes problematic to prawn hatcheries, this Macrobrachium rosenbergii hatchery. The newly hatched larvae, which are devoid of many segments and appendages of the adult, start swimming in about 5 minutes after coming out of the eggshell. At this point of time, the water must be brackish because they are a little bit sensitive to salinity change, and you should take care of what is the requirement of water salinity or water quality by these young larvae. The minute and fragile larvae have no resemblance to spelling mistakes, no resemblance to their parents. So they may not look like what they are supposed to look like as adults. During the entire larval stages, they remain pelagic and actively swim around upside down, grasping their food as they come across. We all know that when they will become adult, they are bottom dwellers. So during this stage, they are pelagic and they will feed on whatever food they come across. They are voracious feeder and will eat continuously as long as food is available. The food consists of zooplankton. In fact, it is always advisable to go for these live food organisms which are preferred by these larvae of Macrobrachium rosenbergii instead of going to supply them some artificial food. They never give that kind of result what we will get from the natural food items. So it's always advisable to go for any of these, some of the zooplanktons, copypods, minute protozoans, rotifers, cyclops, or maybe pieces of animal flesh or food grains or fruits, etc. It depends on you will have to standardize everything according to your own requirement, your own hatchery, your own resources, your own conditions of that place. Now, what are different stages of larval stages of Macrobrachium rosenbergii, the giant freshwater prawn? We have 11 stages: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. And what are the characteristics they have, and which stage comes after how many days of hatching? That we can see here. In stage 1, they have sessile eyes 1 to 2 days after hatching. In stage 2, these Macrobrachium rosenbergii larvae have stalked eyes, and that this stage comes 2 to 4 days after hatching. Then uropods appear at third stage of the larvae, and it is about 4 to 7 days after hatching. Then we have a fourth stage where 2 dorsal epigastric teeth at the base of the rostrum appear, and this stage comes 7 to 12 days after hatching. Then telson becomes narrower and elongated. This is stage 5, and it comes 11 to 16 days after hatching. Then we have stage 6, pleopod buds appear. This comes 15 to 21 days after hatching. Then we have stage 7, pleopods, biramous and bare appear, and this stage comes about 18 to 24 days after hatching. Then we have stage 8, where pleopods with CT appear, and this comes 22 to 28 days after hatching. Stage 9 is characterized by endopods of pleopods with appendices interni, and this stage 9 comes at 25 to 31 days after hatching. Stage 10, the larvae shows 3 or 4 dorsal teeth on rostrum. This stage 10 comes at 28 to 33 days after hatching. Then we have stage 11, teeth on half of the upper dorsal margin, and this may come between 31 to 50 days after hatching. So these are the characteristics on the basis of which we can monitor the progress and development of larval stages of Macrobrachium rosenbergii. By observing these characteristics, we can be sure whether the larval development is showing progress as per the reference which is available with us, whether they are showing normal development, good growth pattern or not. So that can be done, and if there is any kind of deviation from the normal reference, then we can have corrections at that point of time only. Now, if you want to characterize it structure wise, that where and what kind of characteristics are visible at different stages. For example, in a stage 1, what you have to monitor, in fact, is only the eyes. In stage 1, sessile eyes are present. In stage 2, stalked eyes are present. There are no other characteristics which are written here. In stage 3, we can observe rostrum as well as uropod, 2 structures. In rostrum, in stage 3, there is 1 dorsal tooth. In uropod, uropod first appears in stage 3. Stage 4, rostrum has 2 dorsal teeth. So that is how stage 3 and 4 will be distinguished. And uropod becomes biramous with CT. So again, uropod of stage 3 and stage 4 can be distinguished. Stage 5, now we have to see antennal flagellum and telson. Antennal flagellum at stage 5 will have 2 or 3 segments, and telson more elongated and narrower in case of stage 5. Stage 6, we have antennal flagellum 4 segmented, and the telson becomes more narrower than earlier. And the pleopods, first appearance of buds of pleopods, it is at stage 6 that pleopods start showing appearance. Stage 7, antennal flagellum has 5 segments. Pleopods become biramous and bare. Stage 8, about 7 segments appear in rostrum, and then pleopods biramous with CT. Stage 9, about 9 segments in the antennal flagellum, and pleopods, endopods with appendices interni appear. Stage 10, 3 or more dorsal teeth in case of rostrum, and about 12 segments in antennal flagellum in stage 10, and periopods, first and second fully chilated at stage 10. Stage 11, many dorsal teeth on the rostrum, about 15 segments are there on antennal flagellum, and post larvae is characterized by rostrum having dorsal and ventral teeth, behavior predominantly benthic as in case of adults. So a beautiful distinction of characteristics of different body parts in order to distinguish different stages of Macrobrachium rosenbergii which becomes very useful, of immense utility if you are running a hatchery. These are photographs of these stages that you can correlate with the characteristics. Then we have post-larvae. The larval transformation to post-larval stage also marks the end of their life in brackish water, because now post-larvae will come to fresh water. They will remain there until they are adult. They have to go for breeding. Until they are not ready for breeding, they will live in fresh water only. Teeth on upper and lower margin of rostrum. I told this characteristic in the previous slide also. They are generally transparent at this stage and have slightly brown colored chromatophores on the head. Post-larvae exhibit good tolerance to a wide range of salinities, which is a characteristic of freshwater prawns. I told you in the beginning also and the biology also that these prawns, the success of their distribution to variety of habitats, diverse regions, is because of being urohaline nature, the capability to tolerate wide range of salinity variation. They swim against swift currents by tightly crawling on the bottom. They can migrate up to inland lakes, dams, passing along sides, edges, embankments of these streams. So they are bottom dwellers and they can move to different freshwater habitats. During flood seasons, they further move upward to rice fields, small lagoons, and freshwater ponds which are not accessible during dry season. If they get water channel to move, so they can move to any kind of diverse freshwater habitat where they can feed, grow, and mature. From the beginning of their upstream migration up to their adulthood, the prawns stay in freshwater as their permanent home, permanent abode. Adults of these Macrobrachium rosenbergii, the giant freshwater prawn, attain sexual maturity in 6 to 12 months. And how these male and females are distinguished: the second pair of periopods is quite long and has many spines in case of males. Second pair of periopods is quite long. That is one of the very important characteristics, and has many spines. Female, in case of females, the second pair is not so long and is spineless. So when you have to deal with the male and female of Macrobrachium rosenbergii, no issues. You can very easily identify. I have seen these mature Macrobrachium rosenbergii males and females. It's always very easy to distinguish. The genital pores situated at the base of the fifth periopod in case of males, whereas genital pores in case of females are situated at the base of third periopods. Appendix masculina is situated in the second abdominal appendage, males, and maximum size in case of males is 33 centimeter, whereas in case of females it is 29 centimeters. I told you that males in case of Macrobrachium rosenbergii, when they are fully grown, they are larger than the female. So that is how you can distinguish between male and female freshwater prawn on the basis of observation of these characteristics. Presence or absence of the protrusion in the ventral part of the first abdominal segment. In fact, what I suggest you don't have to go through all these details in order to distinguish the sexes. In the previous slide, what I told you, the second pair of periopods, just by looking at the second pair of periopods, if they are long, they are in fact quite distinguishable. They are very long compared to the female, and they have many spines in case of males. So if the second pair of periopods has spines, many spines, and it is long, then it is male. And in case of females, it is not so long and is spineless. So the absence of a spine only you can have that thing. So that's all in the biology of giant freshwater prawn Macrobrachium rosenbergii. In the next lecture, I shall be dealing with the culture method of Macrobrachium rosenbergii. Thank you so much.